Garrison with the University of Washington and University of Washington Botanic Gardens in particular. And I will be speaking on the susceptibility of rhododendron to the azalea lace bug. There we go. The azalea lace bug, Stephanitis pyrioides, is one of the most serious insect pests of the genus Rhododendron, especially azaleas. It is native to Japan and was first reported in the United States and New Jersey in 1916. It was most likely introduced into in nursery stock, as low populations are extremely difficult to see and cause low damage. It has since spread all along the eastern and western North America, wherever rhododendrons are grown. All life stages of azalea lacebug are found on the underside of the leaf. Eggs are the overwintering stage, and eggs are inserted inside the leaf, usually along the midrib. They hatch in the spring and go through 5 instars, or nymph life stages. Early instars are nearly colorless at hatching, and they turn darker after each successive molt, and they get more spikes. Adults have transparent lace pattern wings and pronotum, which are the origin of its common name. The adults then mate, and females insert the eggs into the leaf again and cover them with frass, which hardens to disguise and protect the egg. It can have multiple generations per year until diapause, the trigger for which is currently unknown. It has 2 to 4 generations per year, depending on the climate of the area. Azalea lacebugs cause feeding injury by inserting their stylet, which all bugs have piercing, sucking mouth parts, and they use that to insert their stylet through the stomata and remove all the chlorophyll from the upper palisade parenchyma. This causes chlorotic stippling across the leaves, and severe infestations can result in bleach-colored leaves and eventual plant death. Azalea lacebug feeds on rhododendron and a few other species in the family Ericaceae. Rhododendron is the largest genus in the family, with over 1,000 morphologically diverse species. Displayed here is the current distribution and species richness of rhododendron. It's widely cultivated in temperate regions and is important commercially and ecologically. And that is true in the region where I live currently. This study was done at the Washington Park Arboretum in Seattle, Washington. This park was officially set aside as a botanical garden and arboretum in 1924. And planting began in 1936. The plant collection includes over 14,000 accession specimens, which serve as vital resources for scientific study. The Washington Park Arboretum is part of the University of Washington Botanic Gardens. The maritime climate of the Pacific Northwest is particularly well suited to growing rhododendrons, and most species, except the tropical Variea species, can be grown outdoors, although I can attest we'll try it, but without much success so far. It has several gardens dedicated to rhododendrons, including Azalea Way, the Puget Sound Rhododendron Hybridizers Garden, and Rhododendron Glen. The Arboretum has records for over 4,500 rhododendron plants, making it an ideal location to study this insect and its host. And rhododendron were planted early and often in the Arboretum. So for my, I did this work for my master's degree. In this study, I focused on 2 objectives. The first objective I collected was the seasonality of Azalea lacebug in Western Washington. I collected rhododendron leaves in 2018 and 2019 and counted all life stages. I looked at this data to determine the life cycle timing and developed a predictive degree day model. And I'll only cover this briefly today. I'll be focusing on the second objective, which was to quantify the susceptibility of rhododendron to Azalea lacebug. In this work, I collected and scanned rhododendron leaves from different species and measured the damage. We then looked at this data to see if there was a pattern in the susceptibility across the rhododendron family tree. So in the first objective, 3 rhododendron plants were selected at 6 different sites within the Arboretum, and 216 leaf samples overall from 18 plants were collected 1 to 2 times per week from April to October. So that was mainly focused to get before egg hatch and then to try to see when they kind of trailed off. Azalea lacebug eggs, nymphs, and adults were counted. So some brief highlights from this study. The seasonality study, we observed 2 peaks of early and late instars and a subsequent appearance of adult lacebugs. And we determined that Azalea lacebug has 2 full generations and a partial 3rd generation in Western Washington. We took this count data and developed a predictive degree day model. Presented here on the right is the predicted proportion development of instars and adults and the observed proportional data. So we use this model to predict the degree day accumulation at which certain life stages would occur and identified the most effective control window, which was after egg hatch and before the first adults appear, because the immature forms are the most susceptible to the to pesticides, and once adults appear, they're off laying eggs on new plants, and they're very difficult to control. So to determine a practical recommendation from this model, which is what we're always trying to do, we use climate data from the previous 5 years to look at where this control window falls in a normal year. So in a normal year, the control window is from May 20th to June 3rd, shaded here in green. But in a warmer year, they would develop earlier and could show up as early as May 6th. And in a colder year, they would appear later, and the window could extend as far off as into mid-June. So objective 2, susceptibility of different rhododendron species to Azalea lacebug has been observed, but only studied in a narrow range of species. So to measure susceptibility, we collected leaves from 71 different rhododendron species and cultivars in the August of 2018 and 2019. Leaves were scanned, collected, and then scanned in the lab and analyzed to determine the proportion of leaf damage. And then we looked at the damage proportion across the rhododendron phylogeny and different levels of taxonomy. So previous studies of rhododendron phylogeny and the current taxonomic consensus were used to select from available species and cultivars within the Washington Park Arboretum. So I tried to collect as many species as we had from as wide, as many different clades and sections as I was able. So 80 leaves were collected from each plant over both years, and over 11,000 leaves were collected in total. Leaves were taken immediately to the lab and scanned, and the percent area damage was calculated using Assess 2.0 image analysis software. So this, here you can kind of see at the bottom, the process where the damaged area is highlighted, and then compared to the total area, and the percent area damaged is calculated. This software was originally designed for leaf diseases, but it can quantify the chlorotic area of the leaf. So we started our analysis at the most basic level between those with trichomes, lepidote species, and those without. So when we compared lepidote versus elepidote species, the proportion of leaf area damaged was not significantly different. However, so the presence of trichome structures themselves does not predict susceptibility to Azalea lacebug, but we didn't actually look at the trichome structure of each species, which is very diverse and could have an effect on susceptibility. So that's something further to look into. So then we looked at the subgenus level, Looking at the percent of leaf area damage, you can see that subgenus Azaleastrum, clade C, is the most susceptible to Azalea lacebug. It had the highest overall proportion of damaged, and all samples were observed to have some level of damage. Subgenus Rhododendron was moderately susceptible, with some species showing no damage or very little damage, and some showing a high proportion of damage. And subgenus Hymenanthes, clade B, was the least susceptible, and most species sampled had little damage, and those that did had a relatively low level. Looking at the box plot, you can see that all the subgenera had were significantly different. Subgenus Azaleastrum and Hymenanthes, both lepidote, were observed to have the highest and lowest levels of damage by Azalea lacebug respectively, which is probably why just the presence of scales doesn't predict susceptibility. And the rhododendron, subgenus rhododendron, was between the two. Then I took a look at the section level a little below. The proportion of leaf area damaged to the evergreen azaleas was significantly higher than all the other sections, except for Asian deciduous azaleas, which is in the same subgenus. All the other sections analyzed were not significantly different. Right, so our recommendation is to avoid planting susceptible species, for example, evergreen azaleas, and plant resistant species or varieties instead. But we acknowledge that it's not really possible or practical, as azaleas are commonly a part of historic and culturally important landscapes. These are two from photos from Azalea Way, which obviously prominently features evergreen and deciduous azaleas. So to grow azaleas, an IPM program for the Azalea lacebug will be necessary. And in a public garden, in any garden, our goal in IPM is to protect the public, protect our staff from exposure, and to protect collections. We seek to use the most current science to use the safest, most effective, and least damaging ecologically pesticides as possible. So as it turned out, susceptibility can also have an effect on control recommendations as well. So the plants in Objective 1, the seasonality study, were also collected and scanned in the susceptibility study, and the resistant and susceptible plants were grouped and their development rates were analyzed. So we found that early instar development was not significantly different. So I haven't put up a chart for that. But adult appearance was significant. So degree day requirements for mean adult development in susceptible rhododendrons was 348 as opposed to 382 for resistance. Therefore, Azalea lacebug could reach adulthood up to a week earlier on susceptible rhododendrons than those feeding on resistant ones, shortening the window for effective control. Great. So finally, approximately 70% of rhododendron species are classified as vulnerable, threatened, or endangered, or critically endangered. And 25% of all rhododendron are under threat of extinction in the wild. So identification of susceptible species through research in public gardens will allow susceptible populations to be scouted more intensively for Azalea lacebug and to take measures to mitigate the introduction of this pest into areas in which rhododendron species are endemic. So the susceptibility study is published in the Journal of Environmental Horticulture, and the seasonality study is published in the Journal of Economic Entomology, if you're interested in more detail on those. And so for some future work on rhododendrons at the University of Washington Botanic Gardens, more species, I'd love to evaluate more species for susceptibility to Azalea lacebug to kind of add to our data. There's also it's important to look at alternate hosts of Azalea lacebug. There are several other species in Ericaceae that have been confirmed. Azalea lacebug has been confirmed to be feeding on, such as salal and mountain laurel. And I'm not aware of any research on whether they can successfully reproduce on these species, but I think that is important, as we can't assume isolated populations of rhododendron are not at risk of Azalea lacebug if it's able to spread via other species in the area. And Azalea sawfly. Azalea sawfly is present at the Botanic Gardens, and I'd love to look a little closer at this pest. I really like sawflies, so I'm very much interested in this one. And with that, thank you. I'd like to thank my funding and the Botanic Gardens and my lab assistants. If you have any questions, feel free to email me or let me know now.
Thank you, Ryan. So we can ask questions in the Q&A or raise your hand. I'm going to start with a question that's an observation. So a hobbyist that I know bred an Ostrinum and an Atlanticum. So two native azaleas. And one of the offspring is absolutely gotten hammered by lace bugs. I mean, that thing draws every lace bug from the whole garden. So I feel like his cross kind of broke up, somehow broke up the defense. Can you speak to anything that might be in your wheelhouse that could help me understand that?
I'm not quite sure the actual mechanism of susceptibility or resistance is not clearly understood. There's definitely some research into chemical defenses, which would definitely, if it was crossed and kind of bred out, the chemical defenses that could certainly explain that.
Yeah, I could see that. The other thing is the one that's susceptible has really large soft leaves. They're larger and softer, so maybe a combination of the volatiles and the chemical defenses, and just that it's a tasty leaf is something that I was thinking of, perhaps.
Yeah, definitely. And yeah, since they insert their stylet through the stomata, yeah, there has been research on the size of stomata, but there might be some mechanism there that's
Oh,
affecting.
that's interesting, because you could have a big leaf if you just had bigger cells, which would then have bigger stomata. I know somebody who's studying cell size in rhododendron and the overall impact, so now I have a direction. Thank you. Excellent. Does anybody else have a question? Great. Thanks, Ryan.
Thank you.
I think we're going to have to talk more because we have a group of Satsuki azaleas that vary in their susceptibility. So we had just a trial where everybody who could get eaten did, and we've got variation there. So maybe we'll have to share. Oh, we got a question. From Madeline Couture, what are the best ecologically safe ways to control lacebug? Great question.
Yeah, so fairly safe pesticides that are available are soaps and oils. So the immature forms of the lacebug are very susceptible to those. The only problem is you have to get it on the underside of the leaf where they are. It's a contact pesticide. So those are the safest ecologically. I know systemics are very effective, but definitely would affect pollinators if you're applying them during flowering.
It sounds like, in a way, what you're advocating, though, is it's the right time to do. What you're looking for is the right time to control them, which may be more important because you can use something like a soap if you get them at the right time, yes?
Yes, correct. Yep, there's kind of a narrow window after egg hatch, because the eggs aren't terribly affected by the pesticides, and before the adults appear. because they'll just fly away.
So you have to treat them before you know you have them.
Pretty
Wait until the
much.
next year.
Yeah, yeah, yeah. So yeah, careful scouting during the winter, see that you can't find the eggs.
Nice. Awesome. Thank you.
Thank you.
All right, we've got a couple more minutes to our next talk. Ryan, do you want to stop? Oh, you got it.